Hi kids, welcome to the session. Am I audible and visible to you guys? Hello everyone. Yes, okay. So I'm audible and visible to you guys. Welcome everyone. Good evening. How are you guys doing? How's the day going on? Sorry for the delay. Yesterday I was not feeling well and because of which we have to cancel the session, and today we are going to have your session. In today's session we are going to start with a very amazing chapter, that is your genetics, right? Mendelian genetics, which is a very nice, interesting topic and having a high weightage in your NEET examination. It's a very important chapter. In today's session, we are basically going to talk about the introduction to the Mendelian genetics, who was Mendel, what was the work done by him, and how important it is to understand the genetics. Along with this, we are going to learn about some important terms and the definitions that are required to understand the inheritance of characters. So welcome to the session everyone. Am I audible and visible to you guys? Quickly give me a thumbs up in the chat box. Okie dokie. Rakul. Thank you, bache. Thank you. Fine. Okay, so I'm audible and visible to you guys properly. So let's get started. Let me give you an idea about how important the chapter is. Chapter is super important. It is going to help you in understand not only your genetics but also in your biotechnology, etc. This is simply, an interesting topic to discuss, that how we resemble so much to our parents, and why is it so that if we are having the similar parents, by each and every sibling is looking little different, right? So we are going to talk about different things. We are going to talk about the blue eyes, right? Aishwarya Rai's blue eyes and Himanshi Mam's brown eyes as well. Why am I having this brown eyes? Yes. So we are going to discuss about all of these things. But before we start this session, let me introduce myself. I'm Himanshi Singh, your botany expert, and rest everything is on your screen. The best NEET biology score produced by Himanshi Mam is 360 out of 360 now after your NEET result. So Vedantu had this amazing NEET result, and I hope you guys have gone through that session where all the toppers came in, and today morning it happened. I hope you guys have already gone through it. Now moving forward kids, let's continue and start with our session today. So this amazing man that you can see over here is your George Mendel. He was a person who did a lot of job in understanding how the genetics is going to take place. It's not like that he was the first person to work upon the genetics. There were lots of scientists who were working upon genetics, but what Mendel did was exceptional, and hence we call him as the father of genetics. He was the first person to work upon the genetics, right? So the father of genetics, he was born in 1822. These information are important because certain questions can be asked based upon Mendel as a quotation of, respect to him, that you should know about him at least few important points. So Mendel was born in 1822 in a small village of Czech Republic, and he was the priest. He was a father, a priest in a church. He was not a scientist, right? He was not a scientist by profession. He was just a simple person. Now this simple person, because of his interest into the genetics, understanding how the characters are going to flow from one individual to the next individual. Why is it so that an elephant is always going to give birth to an elephant? Why a pea plant is always going to form a pea plant, right? Just because of his curiosity, he started performing certain experiments, and now we call him as the father of genetics, right? So small little curiosity can lead to wonders. So he started working upon his, he started working upon the pea plant. Along with this, Mendel was exceptionally good in mathematics. It's like, okay, a biology student, he must not be knowing the mathematics. But he was a person who used both biology and mathematics in understanding the genetics. And you know what? I'm going to tell you how he used the mathematics as well, which will make your problem solving, especially from the Mendelian genetics. What will be the ratio? How many phenotypes will be formed? What will be the ratio? What will be the genotypic ratio? It doesn't matter how difficult and long the question is. You guys will be able to give the answer within seconds by knowing these mathematics that was used by Mendel. This is not something new that Himanshi Mam is going to tell you throughout to remember, right? And if you can remember these terms, like 5763, you have to remember just this one, right? 1857, 1863. So you can remember the years because the question was asked directly on the years from the in which duration he was working upon. Now he started working upon the pea plants for the several generation, and this led to the discovery of the genetics, how the genetics is going to work. I hope everything is clear to my kids. Yes? Okay. So let's move forward. So he was the first scientist to apply mathematics with biology, and Mendel found that individual traits are determined by the discrete factors. Factor is an important term. So right now we know that the characters are controlled by something known as genes that is present in our DNA, right? Genes which is present in the DNA. We know now. We know what is gene, we know what is DNA. When Mendel was working, we were not even aware about the DNA molecule. So how can we know the genes? But he was smart enough to identify that there are some factors that are going to control the characteristics. Why I'm having a straight hair, why someone is having curly hairs, why someone is so bright, and why someone is going to have such dark skin tone. All of this is because of the genes, right? The genes are going to control all these factors, and those factors, he used simple term that is factor. Now we know that factors denote gene. This is very important. Don't forget that Mendel never used the term genes. He always used the term factors, right? So he used the term factors for the unit that is going to inherit the character from the parents to the offspring. In 1866, Mendel published his work in a simple Brno Medical Society organization journal, and finally nothing happened, right? There was no progress. He was working upon the pea plant, and there was some other scientist who just asked him that, okay, you should work upon another plant and see whether it is going to work similarly in the other plants as well. Unfortunately, when Mendel was working upon this plant, he could not get all the similar observations because the plant was showing parthenocarpy. And we know what happens in parthenocarpy. There is no sexual reproduction. So the characters are going to remain same, and then Mendel was like, okay, I wasted so many years working upon this pea plant and nothing makes sense. So he stopped working on whatsoever he was doing, and then finally he died in 1889, I guess. I don't remember the year when Mendel died. But anyways, moving forward, what was the work of the Mendel? What were the things that were discovered by Mendel? So Mendel discovered that laws of inheritance. What is inheritance actually? So I look similar to my parent, I look like my mother, I look like my father, right? The passing of the characters is known as inheritance, right? If I call it in Hindi अगर आप लोगों को समझ में आएगी, विरासत में मिलना, something that you're going to get from your foreparents, right? From your ancestors, something that you're going to get. So inheritance, right? We are going to get all the characters from our parents. He deduced as the genes, which was known as factor, come in a pair. This is known as law of pair factors, right? So they are going to present in a pair. This is why, for example, like having the hair texture. Hair texture is a trait which is controlled by a gene. Hair texture is a trait which is controlled by the genes, and there are lots of different types of traits. Yes, we can see curly hair, we can see straight hair. So these are the two different types, right? They are present in a pair, and their inheritance are as distinct unit from each parent. So every parent is going to provide a unit of character. Mendel tried the segregation. Segregation means nothing but separation. So Mendel tracked the separation of the parental genes and their appearance in the offspring as dominant and recessive trait. Now what is dominant, what is recessive trait, what is this genes, what are traits? I'm going to explain everything. You just have to have a little bit of patience. He recognized the mathematical pattern to the inheritance from one generation to the next generation. This mathematical pattern, I am going to teach you all the mathematical patterns that were used by Mendel to understand the inheritance, and it will make your Mendelian genetics super easy. We expect at least one question every year from the Mendelian genetics, and you guys will be able to solve them within seconds with the help of these Mendelian mathematic patterns that were, observed by him, how the inheritance is going to take place. So we'll discuss about them in the upcoming sessions when we are going to talk about the Mendelian crosses. Now moving forward, here comes the important terms. Now everything is going to become clear whatsoever we were talking till now. So the first term is genetics. What is genetics? Study of the hereditary and the variations of the characters in the offspring. So how the heredity is going to take place, how the variations are going to come, and how the genes are going to move from one generation to the next generation is known as genetics. So what is heredity? First of all, we have to understand what is heredity. So heredity is the resemblance between the offspring and their parent. How similar I am to my mother, how similar I am to my parent, how similar are you to your siblings, right? This is known as heredity. So resemblance between the offspring and the parent is your heredity. Then we have a term known as variation. Do I look exactly similar to my mother? Do I look exactly similar to my father? Or do I look exactly similar to my siblings? The answer is no, because we are having certain variations. Whatsoever is passing from the mother, whatsoever is passing from a father to the offspring is going to show certain changes, and these changes is known as variation, right? And the variations are going to occur because of n number of factors. This is the benefit of the sexual reproduction, right? Why the organism is showing sexual reproduction, requiring so much of energy, so much of time, etc. For the purpose of variation, sexual reproduction is going to allow variation in the offspring, and because of the variation there will be better adjustment into the changing environment, and evolution of the organism will continue to take place. The final term is inheritance. Inheritance is the transmission, the movement of the characters from the parent to its offspring is known as inheritance. Are all the chapters clear to you guys? Arvind, we are going to solve all the questions. We are going to solve the mathematical problems as well, as I have already told you. We are going to do everything. Don't worry. Welcome to the session, everyone. Welcome, my kids. Crystal clear. Fine. So these terms are the basic terms that you should know. What is heredity, what is variation, and inheritance. Now moving forward to the next one. The next thing is your gene. So before we talk about the alleles, I would like to talk about the genes. Genes, we know that they are the unit of inheritance. What is going to move from my parents to my body, right? What character is going to move from one generation to the next generation is gene. This is the unit which is present on the DNA, right? Genes are present on DNA molecule. Present on DNA molecule. Now what our gene is going to control? This gene is going to control your characters. Character, I can call it as a trait, I can call it as a character. Let's call it as character. It controls certain character. And now let's take an example. For example, like let's talk about the eye color. So if I talk about the eye color, what is your eye color? This is controlled by gene, right? This is a character. A character is controlled by gene. What kind of eye color are you going to have? I'm having brown eyes, Ashura eyes having blue eyes. Some of you must be having the gray eyes, green eye, etc. Right? So this eye color is a character which is controlled by gene. And there can be many different traits to it, right? There can be many different traits. Now when I talk about the traits are different variants. For example, like we can see green eye, we can see blue eye, we can see brown eye, we can see black eye, etc. Right? So these are the different traits of a character. These are the different traits of a character. Listen to me carefully. A character is controlled by, let me remove this one. A character is controlled by gene. I hope this color is visible to you guys. Is controlled by gene. For example, like eye color. And a character can have a lot of traits. It can have lot of traits, and these traits are controlled by different forms of gene. Different chemical forms of a gene. And these different chemical forms of the gene is known as allele. This is known as allele. And we call them as alleles. alleles. Is this clear, kids? The term, the difference between genes and an allele, the difference between a character and a trait. This is important. These are the basic terms that the student is going to complete the entire chapter, and they're still confused. If someone asks them, okay, can you please tell me what is the difference between a gene and an allele? They're like, okay, allele is the, different forms of the genes. That's it. But you should know the exact meaning, what exactly the allele is, what exactly a gene is. So gene is going to control a character. For example, like eye color, skin color, hair texture, etc. And a character can have different traits. These traits are controlled by allele. Is this clear to everyone? Cool. Now moving forward, so allele morphs, we can see different forms of the character. So we are going to call them as morph, allele. Allele means allele and morph means the different forms that we can see. So allele morphs. The alternative forms of a gene, we can see two different forms, a dominant character and a recessive character. Now what is a dominant character and what is a recessive character? So the character which is expressed is, just a second, let me clean the board first. Fine. So dominant character, the character which is expressed, it is indicated with the capital letter. Now they are calling it as an expressed character. I would like to explain it as a character which is expressed in every condition. Something which is expressed every time. Something which brings a character every time. If it is present, it will work. If it is present, it will work. If I am going to have the allele for the brown eyes, I am going to have the brown eyes, no matter what is present together, right? We will discuss, I think we should first discuss what is your genes and the different types of conditions, right? The genotype, and then I will talk about the dominant and the recessive trait, okay? So we have the term genotype, the genetic constituent of a character. What kind of genes are present in the body? The entire genotype, right? All the genes controlling, controlling the different characters in an organism is known as genotype. Jitni bhi genes present, all the genes, right? All the genes present inside an individual is its genotype. Now we can have two different conditions, homo and hetero. What does the word homo means? Same, right? So if there is presence of a zygote having the same characters, if there is presence of a zygote having the same characters, right? The condition in which the chromosome pair carries similar alleles, same alleles are present. This is known as homozygous. Homo means same. So a zygote which is going to have the same allele is known as homozygous. So we are going to write it as capital T, small t, capital Y, small y. Now, these alphabet represents a allele. For example, like capital T is coding for tallness and small t is coding for dwarfness, right? The short-heightedness. So these are your alleles, right? If there is presence of both T and T, we are homozygous organism, right? We are going to have both two different alleles. So we are homozygous individual having two different alleles. The alleles can be same. Two alleles, they can be same. If they are same, we are homozygous. If they are different, hetero means different. So if there is presence of the heterous condition, different condition is present. For example, like capital T is present with small t, capital Y is present with small y. This is known as heterozygous condition. Are the two conditions clear to you guys? Homozygous and the heterozygous condition. Quickly give me a thumbs up. Yes, that's amazing. In case of any doubts, make sure that you guys are going to ask your doubts as well. Now, what is going to happen? Okie dokie, ma'am. We can see that there is presence of capital T and capital T. We know that capital T is coding for the tallness. Fine, the organism is going to be tall. If there is presence of small t, we know that the organism will be dwarf. But what will happen if capital T and small t is present together? What will happen in this condition? In this condition, the character that we are going to see is tallness. And this is why capital T is dominant. So a dominant character is one that is expressed in each and every condition. It doesn't matter whether it is present in the homozygous condition or the heterozygous condition. It will always get expressed, right? If a character is expressed in both the condition, homozygous and heterozygous, we are going to call it as a dominant allele, simple. And the one that is expressed only in the homozygous condition is known as recessive, right? It is known as recessive. Is this clear to my bacha log? Yes, give me a thumbs up. So we can write it as the character which is expressed in both homozygous and heterozygous condition. Yes, a character which is expressed in both homozygous and the heterozygous condition is known as dominant trait. For example, like capital T, if it is present in capital T, tallness. If it is present in capital T, small t, tallness, homo and hetero condition. Recessive character, the character which is suppressed and expressed, and its character is expressed only in homozygous condition. For example, like small t. What are we going to see when small t is present together? We are going to see dwarfness. Yes, we are going to see a dwarf individual. Clear to my bacha log? These are important terms. How the filial traits are observed in the humans and the plant. This is what the entire chapter is about, right? We are going to talk about them in detail. Now moving forward to the next thing. The next thing is phenotype, the visible characters, the physical expression of the individual. The genotype is going to control the phenotype. So all the genes are going to control what you can see in front of you, right? This is my phenotype. Having different morphological character, having different physiology and anatomy as well. Now we have another term, that is true breeding plant. It is very important. Listen to me carefully. A true breeding plant is the one that produces similar offspring every time, right? They are going to produce similar offspring, which means having similar genotype, and because of the similar genotype, they are going to have similar phenotype as well. Similar to the parent, right? So they are going to have the similar genotype and the similar phenotype every time whenever the cross is going to take place. And this is going to happen because of the homozygous allele. They are going to have the homozygous allele for each and every character. This is known as a true breeding plant. So a true breeding plant is a parent plant that produces the similar offsprings every time due to homozygous alleles, right? Due to homozygous allele. Now moving forward to the next one. The next thing that we are going to talk about is the Mendel's experiment, how Mendel conducted his experiment. So Mendel was working upon the pea plant. The scientific name is Pisum Sativum. So remember the scientific name, and you guys are going to remember it anyways because we are going to discuss it a lot of time. So Pisum Sativum, a pea variety was used. The pea was a small plant and easy to grow. Now why Mendel chose pea plant? Why not a mango tree? Mango are so tasty to eat, right? Or why not a religious plant like people or anything else? He chose pea because of certain reasons. Now what are the reasons behind it? It is small to grow. We can grow this plant even in a small area. Along with this, it is easy to grow. It is self-fertilizing plant, right? It does not show cross-pollination naturally. But if we want, we can do a cross-pollination very easily. It can undergo the cross-pollination if we want, right? So we can do the experiment and we can easily make the plant make a cross between the plant. And they have the short lifespan, which means in a shorter span, I can get the offspring. If I talk about the humans, we have the gestation period of 9 months. Not only this, the person will hit the puberty, attain the sexual maturity, and then the offsprings will be produced, which means it is going to require at least 16 to 17 years, right? Minimum of 16 to 17 years, and which is not easy to work with. So we want to have a plant, or we want to have an individual or organism that is going to have a shorter lifespan. They're going to have a shorter lifespan, and in this shorter lifespan, they're going to produce lots of progenies at every time. The larger the number of the progenies is, the better is the results, right? We can easily evaluate the result. Whenever you are going for the scientific studies, the career is going to change. Not everyone is going to become the doctor. It is the harsh reality, and we have to face this thing. But yes, when you are going to move out into the different industries, some of you might enter into the scientific work. And let me tell you, whenever we are working with the science, we need to perform the experiment n number of times so that we have a lot of quality. The more results we have, the more appropriate the result is considered as, right? If I'm going to perform something at once, yeah, it's like, flipping a bottle, right? If you want to flip a bottle, you can flip it in the one try, you cannot flip it in a one try. It's not like, okay, I did it in the first try, I can do it every time. No, right? So we have to do a lot of qualitative analysis. So next generation can be studied in the small duration, and not only this, the next generation is produced in many number. Higher number will be present, the better the result will be. So smaller lifespan. There are three basic characters that are very, very important. Self-pollination is shown. Next thing, we can perform the cross-pollination if we want. Another thing is the shorter lifespan along with lots of progenies. High number of the progenies was produced. Now moving forward to the next character, Mendel selected 7 pair of true breeding plants, right? 7 pairs of the true breeding garden pea plants, Pisum Sativum. So he used 7 different characters. 7 characters were studied by Mendel. Now, flower color, they can be presence of the violet flower, they can be presence of the white flower, the plant height, they can be tall, they can be dwarf. Seed color, it can be green, it can be yellow. The seed can be wrinkled, the seed can be swollen, right? It can be round. The pod color, how is the food color, right? The fruit color, the pod color is going to be either green or it is going to be yellow, right? It can have a inflated shape or it can have a constricted shape. Inflated means that it is going to have a proper, you guys must have eaten pea, right? So this kind of shape that you see is known as the inflated shape, and another shape where you can count the number of the peas inside is known as constricted shape, right? This is the constricted shape. And then finally the flower position, axile and terminal. Now when we are talking about a character, so all of these are the characters that are controlled by the genes, and these are the different traits that are controlled by the different alleles. Now you guys can understand a character and a trait. You can understand the genes and the alleles. Now we can have a dominant form and a recessive form. It is very important to remember the dominant and the recessive form. So stem height, tallness is dominant, recessive is dwarf, which means tallness will be expressed in both the condition, homozygous and heterozygous, if capital T allele is present. Purple color is dominant, axial position is dominant, which means we have this plant. What is the axial and the terminal position? We have this plant, right? If the flower is present on the apex, this is known as terminal position. And if the flower is present on the sideways, then this is known as axial condition. Axial condition is more dominant. Inflated, having a swollen pod. Constricted, where you can count the number of peas inside. The green pod color is dominant along with the yellow. Seed shape, round and wrinkled. And another one that is very, very confusing and the students always make an error is the seed color, yellow is dominant. Yellow seed color is dominant. So whenever you are going to eat the pea, yes, whenever you are going to eat the pea, we can see the green color all around us. So the pod color, the dominant pod color is green, but the dominant seed color is yellow. Please remember this point. It is slightly pale in color and we consider it as yellow. Very, very, very important. So you can remember it like, in a related manner. I remember it like this only. That the pod colors are green. Whenever I'm going to eat the pea, they are green, green color, right? So green pod color is dominant. But if I talk about the seeds, yellow color is the dominant form, right? Yellow color is the dominant form. Is this clear, bacha log? Yes? Give me a thumbs up. Please remember all of them. I think they are easy to remember. All you have to do is, just go through them once, twice, or solve certain questions, and you will remember the dominant and the recessive characters. What is more important to remember is the last character, that is your seed color, because this is where you're going to make the error, and the examiner knows this well. So this is very, very important. Cool. Now let's move forward to the next one. How Mendel did his experiments. So we know that whenever we are going to make a cross, we need to select two parents. So he always selected a pure breeding plant, right? Pure breeding plant. I hope you guys remember what is the definition of the pure breeding plant. So he selected two pea plants with contrasting character. Now when he was using the contrasting character, he was looking only after one character. Either I am going to look after the flower color, or I am going to look after the plant height. If I am using the plant height, I am only going to look after, okay, the plant is tall, the plant is dwarf. That's it. It doesn't matter what kind of seeds are present, what kind of flowers are present, what is the flower position, nothing matters. What matters is only the plant height. If I'm going to look after the flower color, oh, whether the flower color is violet or white, yes, this is what matters. Nothing else matters. Only one character at a time, right? And he was using the pure breeding plant, which means that they were going to have the homozygous condition. So two pea plants were selected. Removal of the anthers, emasculation, and the bagging technique was done. Let's understand what is emasculation and the bagging technique. So here comes your emasculation and the bagging technique. So first of all, what is going to happen? This pea plant is a bisexual plant having a papilionaceous corolla. Because of the papilionaceous corolla, this standard is going to cover the entire reproductive structures, and the plant is going to show self-fertilization, right? Self-pollination will be present in the normal condition. Now, emasculation and the bagging technique is done for the cross-pollination. If we want to do the cross-pollination, right, in this condition, we will do this, use this technique. So, there is presence of the carpel, the female part, and there is presence of the stigma, that is your stamen, that is your anthers, the male reproductive part. The first thing is emasculation. E means removal and masculation means man, right? So emasculation, we are going to remove the male part. Emasculation, removal of the male part. So the anthers are removed. We are going to cut and remove the anther before maturity, right? Before maturity, when the flower is very small, we are going to do this emasculation. Now, once the emasculation is done, we want to protect the stigma from the unwanted fertilization. Yes, we need to protect the stigma from the unwanted fertilization, and hence we are going to do the second part, that is bagging. We will do bagging, and this is how your bagging is going to look like. We are going to cover the flower inside a bag. The bag is usually made up of the paper so that the air can come in and go outside, and the flower can easily breathe. So this is the bagging to prevent. Why are we doing bagging? To prevent unwanted pollination. Yes, to prevent unwanted pollination. Once the bagging is done, the flower will mature. Once our stigma is ready for the sexual reproduction, the pollen is obtained from the white flower, or we can call it as a desired flower, right? Whatsoever the desired plant is, वहाँ से हम collect कर लेंगे. We will collect the pollen grains from the desired plant, right? Our character, our important character, right? So we will obtain the pollens from the desired plant, and then finally the pollen is transferred to the purple flower, the emasculated flower. So we have transferred the pollen grains. We are going to dust them. This actual artificial pollination will take place, right? Our desired cross pollination is taking place right now. Once the cross pollination is done, again we are going to do the bagging. Again, bagging will be done to prevent further cross pollination, further undesired pollination to take place, and then finally the progenies will be obtained. Once we are going to obtain the progeny, now we are going to sow them, look after their characters, and make the genotypic and phenotypic ratio. This is how Mendel worked. It was a very tedious job to do, very difficult task to do, especially when we are working with the thousands of progenies at a particular time. So it was a very difficult task, very patience is a lot of patience is required, and he did his work with a quite justice, right? So this is how the emasculation and the bagging technique is going to work. First of all, we have to select the parent. Once the parent is selected, for the desired cross-pollination to take place, we have to do the emasculation. After emasculation, bagging will take place. Once the stigma is reproductive, we are going to do the cross-pollination. We will collect the pollens and we will do the cross-pollination. Once the cross-pollination is done, again bagging will take place and the progenies will be allowed to develop. Once the progenies are developed, then we are going to collect the progenies, sow them and look after their character, determining the inheritance. Is this clear to my kids? Kevin, this is basically for your class 12th, right? It's for your class 12th. Okie dokie. Clear to everyone? Any doubts, any confusion? In case if you guys are going to watch the video later on, you're not live in the session, make sure if you guys have any kind of doubt, you're going to drop your doubts in the comment section, right? And I will revert back to each one of you. All the doubts should be cleared. If we are providing free education, this free education should be quality education as well. Now this is your homework question, my kids. And how many true breeding plants were used by the Mendel in his experiment? It's a very nice question. Read the question carefully. This is your homework question. I want you guys to drop your answer in the comment box after the session. And in the next session, I'm going to give the shootout to the students who are going to give me the correct answer. It's a very nice question. I'm telling you guys. How many true breeding plants were used by the Mendel in his experiment? Right? Read the question carefully and make sure that you guys are going to give me the correct answer in the comment section after the class. And the students are going to watch me later on. Please do make this happen, right? Now moving forward, do you guys face any of these problems? You guys are having lots of doubts, no conceptual clarity. You want to have the personalized attention which is lacking in the YouTube, or your syllabus is lagging behind. You want to have lots of assignments and the questions, etc. You are looking for a revision plan especially for your NEET 2022, and you prefer to learn Hindi over English, or you prefer to learn in English over Hindi. Any language that you want, we are going to teach you in your demanding language, right? So we will teach you in both the languages with the help of the Vedantu's live interactive course. So talking about this Vedantu's live interactive course, this is our session. Oh, I'm here. So you can see that in the description box. In this description box, kids, when you're going to open this description box just below the video, you guys can see that there is presence of lots and lots of links. So many links are present. But if you guys are preparing for your NEET examination, for your CBSE examination, you can click upon the link. You can change the course if you guys want to change it for grade 12 NEET preparation. These are the courses that we are offering you guys. So this is the course that you can see. There are 3 different plans. My favorite one is the classic subscription, where you are going to get everything that is essential, right? So in this classic subscription, kids, it will cost you 41,399 for the entire 12 months. And if you guys want to go for the 1 month subscription, you guys want to go for the 15-year trial, you can go for that as well. So for the 15 days, we are going to give you free classes. You have to pay this amount. In case if you guys don't like the Vedantu classes, you can ask for the complete refund. We will refund all your money back. And if you guys enjoy it, which is going to happen, you can go for the yearly subscription. Now in this classic subscription, we are going to provide you guys with the live online interactive classes. Lots of tests and their analysis will be provided to you, kids. And every test you are going to see your AIR ranking, because there are so many students who are preparing from Vedantu. And you can always look at your performance. You can compare your performance among those different students from the other part of the countries, right? Along with this, analysis is very important because we have to work upon our negative marking. Assignments and notes will be provided. After every session, you will get the detailed notes, handwritten notes of your teachers, assignments, etc. Doubt solving during the class with the help of the master teacher and the class teacher. So master teacher, the one that you can see in front of you, is the master teacher. Along with the master teacher, there will be presence of the class teachers, not only one, but lot of them will be present, who are going to solve all your doubts. So whenever you are going to leave the session, you will be 100% satisfied that yes, my doubts have been solved. Now moving forward, doubt solving on the mobile app is applicable only on the classic subscription. So go for this classic subscription where you are going to get your doubts solved even after the class. So after class, you can ask your doubts to your doubt experts who are going to stay with you kids from 8 a.m. to 11 p.m. This is very, very important, right? Not every time you're going to have a doubt from the topic that is going in the class. You will have the doubts later on as well. And I think majority of doubts come when we are going to revise the chapter on our own, right? So you should have some doubt experts who are going to solve your doubts. So doubt solving on the mobile app. And if you guys want to have a personal mentor, you can go for the plus subscription as well. So these are the 3 different subscriptions that we have, and the prices are very minimum. The price is 1,980 for 15 days, which is completely refundable. You guys can calculate the price per month, per day, how much it is going to cost, and it is, you know, buying a chocolate every day. It's as cheap as buying a chocolate every day, right? 10 rupees chocolate, 15 rupees chocolate every day. That's it. Now don't forget to use this special sorry. Okay. Now I'm fine. So moving forward, kids, don't forget to use this special discount code HSPRO to avail your 10% off, right? On all the prices, you can get additional 10% off by using this special discount code. So the link is in the description box. You can go for the CBSE, NEET, etc. This is about the Vedantu's life course for your entire syllabus. Don't forget to subscribe to our VLE Telegram group. Don't forget to solve your homework question, and don't forget to hit the thumbs up button, share the video, and subscribe to subscribe to the channel if you haven't subscribed yet. Till then, bye-bye, take care, and always keep on smiling. So thank you, kids. Don't forget to answer the homework question in the comment section. And along with this, don't forget to ask your doubts as well. Till then, bye-bye, everyone. Always keep on smiling, and don't forget to read your NCERT. I will see you guys soon. Ta-da!